A very good afternoon to all. So poll has been launched. Quickly answer through poll. My audio is clear and my screen is visible. So students, how's your preparation going? कैसी चल रही है आप सब लोगों की तैयारी for NEET exam? Okay. So don't worry, don't stress yourself. I'm pretty sure that you all are going to secure a very good rank in NEET, okay? So stay positive. So let's start with today's lecture. Today we are going to discuss molecular basis of inheritance, part 2. Earlier, in the previous class, we have discussed replication. We have discussed the structure of polynucleotide chain, the structure of the DNA, and the discovery regarding the genetic material. So today we are going to discuss transcription, translation, and regulation of gene. These are very important, okay? So transcription is the copying of information from the DNA to mRNA. This process is known as transcription. For example, this is your DNA, and this DNA, we know that the DNA is a double-stranded structure, right? It has two strands, and both the strands, they have antipolarity. They have opposite polarity. Like one strand is 3' to 5', and another strand is 5' to 3'. So they have opposite polarity. During transcription, we use 3' to 5', and the strand which is having a polarity 3' to 5', and we call this strand as a template strand. So the mRNA uses the RNA polymerase, which is a DNA-dependent RNA polymerase enzyme. Why it is DNA-dependent? Because it is using the one strand of the DNA as a template for the synthesis of the mRNA. So it is a DNA-dependent RNA polymerase. So the RNA polymerase will use this 3' to 5' end of the DNA as a template and will synthesize the mRNA. This process, where the genetic information is transferring from the DNA to mRNA, this is known as transcription. Okay, and this mRNA and the information when transferred from the mRNA to protein, this process is known as translation. Here ribosome play a very important role. And remember one more important point, that we know that mostly the viruses, they have RNA as a genetic material, right? So the process where the mRNA is converting to tRNA, that process is known as reverse transcription. Okay, you can see this. There's a reverse process is taking place. That is why it is known as reverse transcription. In generally, the transcription is from DNA to mRNA formation. But if we are forming the DNA from the mRNA, then it is known as reverse transcription. And in this process, the enzyme involved is known as reverse transcriptase enzyme. Okay, remember this. And one more important point, you can see that DNA, it replicates by this replication. The replication is taking place, this transcription is taking place, this translation is taking place. So replication, transcription, translation, they entirely called as central dogma of biology. This entire process of which includes replication, transcription, and translation, they are referred as a central dogma of biology. Moving next is the transcription unit. So transcription unit, it consists of three components. Number one is the promoter. This is the promoter, where the RNA polymerase will come and bind and initiate the transcription. Second one is the structural gene. And the third one is the terminator. Terminator where the transcription stopped. Okay, so that component is known as terminator. And also one important point that the position of the promoter and the terminator, they are defined with respect to the coding strand. You can see this, the template strand, they have a polarity 3' to 5', and the coding strand, they have a polarity 5' to 3'. Okay, so we always say that the promoter, they are present upstream. Okay, they are always present upstream of the structural gene, or they are present at the 5' end upstream of the structural gene. So that is the position or the location of promoter. Similarly, for terminator, we said that the terminator, they are present downstream. So this is downstream. So terminator, they are present at the 3' end downstream of the structural gene. Okay. Remember this, that we are defining the position of promoter and terminator with respect to the coding strand, with respect to the polarity of the coding strand. Okay. Now we know that the gene is a functional unit of inheritance, right? There's a term called cistron. Cistron is a segment of DNA which codes for a functional protein, or which codes for a polypeptide. So in eukaryote, we observe monocistronic. The structure genes are monocistronic. Mono means one. You can see this, here's one gene. So it's monocistronic. And this P represent promoter and the T represent terminator. In eukaryote, always remember that every gene has a specific promoter. They have a specific promoter and specific terminator. Remember this, that in eukaryote, every gene will have their specific promoter. But in prokaryotes, they have polycistronic. Poly means many. There are many cistrona present like this. Here's gene 1, gene 2, and gene 3 is present. And there's a single promoter. Okay, so in prokaryote, you will observe that one promoter can transcribe many genes. You must have learned about this lac operon, right? Lac operon also has the same case. They also show the condition which is polycistronic, like they have a lac promoter and they have 3 structural genes. So there's a single promoter, which is the lac promoter, and it is transcribing the 3 structural gene, which is Y, Z, and A in case of lac operon. So prokaryotes, they are polycistronic in nature, and eukaryote, they are monocistronic in nature. Okay, remember this. Next, let's discuss the transcription. So transcription in prokaryotes and eukaryote differ. Okay. In prokaryotes, we know that the prokaryote, they do not have a well-defined nucleus because they lack nuclear membrane, right? So transcription in prokaryotes, it occurs in cytoplasm. Remember this, in transcription in prokaryotes, it occurs in cytoplasm. And in prokaryotes, this RNA polymerase is present, right? This RNA polymerase, there's a single RNA polymerase which can synthesize all the RNA. That this single RNA polymerase can synthesize rRNA. It can also synthesize tRNA. It can also synthesize mRNA. So there's a single RNA polymerase can synthesize all the RNA. Okay, and in case of prokaryotes, this RNA polymerase, it made up of 5 subunits. And what are those subunits? Number 1 is alpha, there's beta, a beta prime, there's omega, and last one is sigma. Remember this, that these 4 subunits, they form the core enzyme. And this sigma factor, when this core enzyme and the sigma factor combine, they form the holoenzyme. Okay, what is the main function of sigma factor? Sigma factor role is in the recognition of the promoter, because we know that the transcription cannot occur at any place in the DNA. It has to be occur at a specific site. Now, who will going to recognize that site? That site will be recognized by the sigma factor. Sigma factor provide the specificity to the RNA polymerase. Sigma factor will recognize this promoter region, because every promoter, they have a conserved sequences. Like, for example, if I take the bacteria from different places, okay, but each bacteria, the promoter, they will have the conserved sequences. Like whether it's a bacteria from for example, let's take Jammu, whether it's a bacteria from Jammu, whether it's a bacteria from Kerala, whether it's bacteria from Uttar Pradesh, they're from different state. I obtained this bacteria, but every bacteria will have the same conserved sequences, right? And who will recognize these conserved sequences which are present in the promoter? They will be recognized by this sigma factor. So sigma factor will recognize these conserved sequences, and it will allow the RNA polymerase to bind to this promoter. So when the RNA polymerase bind to the promoter, then it will synthesize the RNA chain. Okay, because the main function of RNA polymerase is to synthesize the RNA chain. So it will synthesize the RNA chain, which is approximately 8 to 9 nucleotide. When the RNA polymerase synthesize a RNA chain of 8 to 9 nucleotide, then the sigma factor get released, because the role of the sigma factor is over, because the role of the sigma factor is just to recognize the site where the transcription takes place, just to recognize the promoter. Okay, just to recognize the location of the promoter, so that the RNA polymerase can bind. But once the role of the sigma factor is completed, the sigma factor will get released. And now the beta and the beta prime, they will play an important role, because the beta and the beta prime, they will act as a catalytic center. Their function is in the elongation. Now they will keep on adding the nucleotide in the RNA chain, and they will increase the length of the RNA chain. Now the last step is the termination. In prokaryote, there's a factor called Rho factor. So the Rho factor will bind to the RNA polymerase, and it will cleave the RNA DNA hybrid. So once it reaches the RNA DNA hybrid, this Rho factor will cleave the RNA, and the RNA will be released from the DNA. So this is a newly synthesized RNA. And always remember, in case of prokaryotes, the transcription and translation both takes place in the cytoplasm. And we know that the RNA, instead of thymine, they have uracil. Remember this. If this is a DNA, it's having a sequence like A, T, G, C, T. So the mRNA form will be, and the synthesis of mRNA will be always in 5' to 3' direction. Always remember this. Whether it's replication, whether it's transcription, the synthesis will always occur in 5' to 3' direction. So this is the mRNA. And the mRNA is complementary to this template strand. So here's A. So we generally write T, but it's the case of mRNA. It's a case of RNA. Instead of T, we will write U. A, C, G, A. Remember that RNA contains uracil instead of thymine. In case of prokaryote, you have observed that the RNA polymerase are directly binding to the promoter, right? And the sigma factor is helping the RNA polymerase to bind to the promoter. But in case of eukaryotes, the general transcription factors are present, because the RNA polymerase, they cannot bind to the promoter directly. They are binding indirectly with the help of the general transcription factor. Now, in case of the eukaryote, the conserved sequences are Tata box. You can see these are the conserved sequences which are same. Okay. So in case of eukaryote, the conserved sequence are Tata box which are present in the promoter. Now this first general transcription factor is TF2D. It will recognize this conserved sequence. Then it will bring TF2B TF2B role is to recruit the F. Okay. Now this TFF will bring this RNA polymerase. Now the RNA polymerase will bind to the promoter. Okay. You don't have to memorize these the name of the general transcription factor. It's just for your knowledge. I'm telling you this. You don't have to memorize this. Please, just for the knowledge and just for the concept, so that the concept will become clear for you, that how this transcription is taking place in eukaryote. That is why I kept this picture. Okay, so don't memorize these factors' name, but just remember this, that in case of prokaryote, the RNA polymerase, they are directly binding to the promoter. Okay, but in case of eukaryote, the RNA polymerase are not directly binding. There are some general transcription factors are present, which are helping the RNA polymerase to bind to the promoter. And in case of eukaryote, remember this, it's very important. In case of eukaryote, that different RNA polymerase are present, like the RNA polymerase 1, the 2, and 3. In case of prokaryote, there's a single RNA polymerase which has synthesized all the RNA. But in case of eukaryote, there are different RNA polymerase are present, like 1 will synthesize rRNA, 2 will synthesize mRNA, and 3 will synthesize tRNA. Okay, remember this. Many times this question came in NEET, okay? And one more important point, when this RNA polymerase, they bind, they will start synthesizing the RNA chain, right? Termination. In case of prokaryote, the termination was taking place by, quickly answer me in the chat box, with that factor which is involved in the termination in case of prokaryotes. Okay, so in case of prokaryotes, just now I've told you that in case of prokaryotes, the termination is done by the Rho factor. Okay. But in case of eukaryote, termination is done by the polyadenylation sequence. Because in mRNA, there's some sequence are present, which are known as polyadenylation sequence. And these sequences are recognized by some factors. They are cleavage stimulating factor, and they are cleavage and polyadenylation specific factor, which recognize this sequence. Once they recognize this sequence, now this enzyme poly-A polymerase, right? This enzyme will keep on adding the poly-A tail at the 3' end. Like this. It keep on adding the poly-A tail at the 3' end, which leads to the termination in case of eukaryote. Okay? And in prokaryote, the transcription is taking place in cytoplasm. In eukaryote, the transcription takes place in nucleus. Remember this. Because eukaryote, they have a well-defined nucleus, they have nuclear membrane, right? Moving next, we know that the prokaryotes, they does not show the post-transcriptional processing event because they does not have introns, they does not have junk DNA, they does not show splicing, okay? But in eukaryotes, we are more complex. The processing is required. And this processing includes capping, tailing, and splicing. In eukaryote, first the mRNA form is known as when the RNA is formed, it is known as a primary transcript. And this, we call this RNA as a heterogeneous RNA or the heteronuclear RNA. Now this RNA will be converted to mRNA when this RNA undergo processing. Once this RNA, primary transcript RNA undergo processing, after that, we get a final transcript which is known as mRNA. So first is the capping. Capping is done at the 5' end. And the enzyme involved in the capping is guanine 7-methyl transferase. Tailing is done at the 3' end. The enzyme involved is poly-A polymerase, which keep on adding the adenine at the 3' end. Now splicing, that is a removal of introns. We know that in eukaryote, if this is a eukaryote, these exons are present and introns are present. Right? Exons are the coding sequence which code for the functional protein, and introns are the non-coding sequence. Okay, and for the formation of the protein or the polypeptide chain, we only require the coding sequence, which does not require the non-coding sequence. So we have to remove this non-coding sequence, and for that we require splicing. And finally, the mRNA final transcript is known as mRNA, and now this mRNA will be transferred from the nucleus into the cytoplasm through the nuclear pore, because the nucleus, they have nuclear pore, right? So this mRNA, after processing, will enter into the cytoplasm for translation. In prokaryote, the transcription and translation is taking place in the cytoplasm, but in eukaryote, transcription takes place in the nucleus and translation occur in the cytoplasm, because the ribosomes, they are present in the cytoplasm. Remember this very important point. And one more important point, that why this capping and tailing is done, right? It's to protect the mRNA. Because in cytoplasm, their exonucleases are present. So we don't want this mRNA to get degraded. So we have to protect this mRNA. So capping and tailing, they provide protection. And also this capping, they are recognized by the ribosomes which are present in the cytoplasm during translation in case of eukaryote. Moving next is the tRNA. tRNA, the other name of tRNA is the adapted tRNA or the transfer tRNA. Okay, and you can see in the image, the tRNA, they have D loop, they have T loop, they have anticodon loop, and they have the acceptor arm. This is the acceptor arm. This is the site or the arm where the amino acid will get attached. Okay, and the anticodon loop, they have the anticodon. They have the sequence which are complementary to the codon which is present in the mRNA. And the secondary structure of tRNA is the clover leaf-like structure, and the tertiary structure, or in the three-dimensional view, the tRNA, they have an inverted L-shaped structure. And every amino acid, they have a specific tRNA. Okay, so each amino acid, there's a specific tRNA present. For stop codon, there's no tRNA. Remember this. For stop codon, there's no tRNA. Now let's discuss the translation. When the information is transferred from the mRNA to the protein, this process is known as translation. And translation is also referred as the process of polymerization of the amino acid to form polypeptide chain. First step of the translation is the charging of tRNA. And the enzyme which is involved in the charging of tRNA, charging means that attachment of the amino acid to the respective tRNA. So the enzyme which is involved in the charging of tRNA is known as amino acyl tRNA synthetase. So this is the enzyme. First, the ATP will bind and the amino acid will bind. And the amino acid AMP complex is formed. When the tRNA will bind, now the amino acid will bind to the acceptor arm of the tRNA, and the tRNA gets charged and the AMP is released. We call it as charging because in this process, the ATP is involved. Okay. And this charged tRNA which is carrying the methionine amino acid, is known as an initiator tRNA. Now let's discuss the translation. In prokaryotes and eukaryotes, they have a ribosome binding site in the mRNA. In the mRNA, there are some sites are present where the ribosome will bind. In case of prokaryotes, these sites are known as Shine-Delgado sequence. These sequences are present in the mRNA. And these are the sequence where the ribosome will come and bind. And we know that the ribosome, they have two subunits. There is large subunit and the small subunit. And this small subunit, it has three sites. There's E site, P site, and A site. Once the smaller subunit of RNA, first remember the smaller subunit of RNA will bind to the ribosome binding site present on the mRNA. Now the small subunit of ribosome will stall over the mRNA. Okay, it will move over the mRNA until it encounters the start codon. And we know that the start codon is AUG. And this AUG performs dual function. It is acting as a start codon and it also codes for methionine amino acid. So this E site is the exit site. The P site is the site where this start codon is present, and this A site, which is the amino acyl site. For the initiation, there are three factors are present in prokaryotes. And these factors are IF1, IF2, and IF3. First, this IF1 will bind to the A site of the small subunit of ribosome. So here binds the IF1. IF3 will bind to the E site. Here IF3 will bind, right? IF2 plus GTP plus charged tRNA. Now let's discuss the function of these factors. IF3 will bind to the E site because it prevents the association of the larger subunit with the smaller subunit. When this IF3 is bind to the E site, it does not allow the larger subunit of the ribosome to bind to the small subunit. So no complete ribosome will form until this IF3 is bind to the E site. IF1 will bind to the A site so that the IF2, which is recruiting this charged tRNA, will bind to the P site because this charged tRNA is carrying the amino acid which is methionine, right? And the start codon is present in the P site. So this charged tRNA has to bind in the P site. So IF2 role is to recruit this charged tRNA. So this charged tRNA will bind in the P site. Okay. Now you can see this. Once this IF2 bind, now this IF3 will get released. When it is released, now the large subunit of ribosome can bind to the small subunit and a complete ribosome will formed. Once this complete ribosome will formed, this GTP get hydrolyzed, which is associated with IF2. Once it get hydrolyzed, now this IF1 will also get released and IF2 will get released. Now the process of elongation will takes place. Now in elongation, you will see this. Now the largest subunit has bind because the IF3 factor has released. Here's the tRNA. This tRNA is having a methionine. The codon here is AUG. Now the another tRNA will come, which is having a, let's say this A site, this is the A site, this is the P site, this is the E site. This A site is having a codon which is GUG, and this GUG will code for another amino acid, right? So this tRNA is carrying the amino acid which is coded by this GUG. Now this tRNA will come and bind to the A site, right? Now this tRNA has bind to the A site. Now the peptide bond formation occur between these two amino acids. And this reaction is catalyzed by an enzyme which is peptidyl transferase. And this enzyme is a part of large subunit. So large subunit role is in the formation of the peptide bond because it has this peptidyl transferase enzyme. So now the peptide bond formation will occur between these two amino acids, and this amino acid will transfer to this tRNA. And this tRNA will shift to they will translocate like this. This will move to the E site, and we know that E site is the exit site. So once this amino acid is transferred to the tRNA, which is present in the A site, this tRNA, which is present in the P site, will move to the E site and it finally get released. What is this image is showing? Okay, it is showing the same phenomena. This tRNA is coming. There's a peptide bond formation is taking place. This complete polypeptide will be transferred to the tRNA, which is present in the A site. And now this tRNA, once it transfers the polypeptide chain, it will leave the ribosome complex. After elongation comes the termination. There are some release factors are present. There's RF1, RF2, and RF3 in case of prokaryote. The role of these release factor is in the recognition of the stop codon. Okay, once they recognize the stop codon, then the translation gets stopped and the ribosome subunit, they get dissociate. And the polypeptide chain is released. So translation in case of eukaryote is differ from the prokaryote. Number one, in prokaryote, there's a 70S ribosome, but eukaryote, they have 80S ribosomes, right? In prokaryotes, they have Shine-Delgado sequence, right? In eukaryote, there's a Kozak sequence are present. In prokaryote, there's a Shine-Delgado sequence are present, which is acting as a ribosome binding site. In case of eukaryote, they have a ribosome binding site, which is a Kozak sequence. And the factors are differ in case of eukaryote. Okay, they have EIF1, EIF2, EIF3, and EIF4G, and so on. You don't have to remember these factors, but just try to just try to understand the concept. Okay, don't memorize these factors. Just try to understand the concept that how this tRNA they are coming and binding, how this amino acid, the peptide bond is forming. Okay, just and try to understand the concept. And in eukaryote, the factor are ERF1 and ERF3. Okay, they also, the role of the ERF3 is not clear, but the role of ERF1 is in the recognition of the stop codon. Moving next is the genetic code. Remember these properties of genetic code. These are very important. We know that there's 64 codon. Out of these 64, 61 code for amino acid and 3 are the stop codon. And this, we know that AUG is the start codon which codes which perform dual function. It codes for methionine, and it also acts as a start codon or the initiator codon. First property of genetic code, that is unambiguous. Unambiguous or non-ambiguous means that each codon codes for a specific amino acid. Degeneracy, as you can see that triple U, there's UUC, they all are coding for phenylalanine. Okay, that means that more than one codon can code for the same amino acids. This is what we call degeneracy. Universal, like, well, for example, this UUU, this codes for phenylalanine in case of bacteria, and it also codes for phenylalanine in case of humans. So it is nearly universal. The genetic code is universal, non-overlapping. For example, if this is a sequence, the 3 nucleotide, they will form a 1 codon, right? This AUG, they are forming a 1 codon. These are forming a 2nd codon. They are forming a 3rd codon, right? But there should be no overlapping. This G is part of the codon number 1. Okay, this G is part of codon number 2. This C is part of codon number 3. It cannot be like this. If it is AUG, it cannot be. It cannot be like this, like UGG. They should not be overlapping because this G is a part of 2nd codon. It cannot be a part of the 1st codon. Okay, there should be no overlapping. Now, regulation of the gene expression. In eukaryote, the regulation, it could be at the transcriptional level, at the processing level, at the translational level, and when the transport of mRNA from the nucleus to cytoplasm. Remember that where the regulation is taking place in case of eukaryote. These are very important. You don't have to go in detail because it's not given in NCERT. So just remember these levels, the transcription level, processing level, and the transport of mRNA from the nucleus to cytoplasm, the translational level. These are the 4 steps where the regulation takes place in case of eukaryote. Now let's discuss the lac operon. This is a case of polycistronic. Remember this. Because there's a single promoter, which is a lac promoter, and it is transcribing all the structural gene, which is ZYA. This operon model was observed by Jacob and Monod. The operon model, it consists of structural gene and the regulatory genes. This I gene is the regulatory gene. This I gene will code for the mRNA. This is the promoter of the I gene. Now this mRNA, when it is translated, it forms a protein which is the repressor. We know that the repressor will suppress the transcription. Okay, so repressor lead to negative regulation because it is suppressing the transcription. And if there's an activator is present, then it will lead to positive regulation. Because the activator role is to promote the transcription. So it will act as a positive regulation. So in case of lac operon, the inducer is lactose. Remember the inducer, that inducer is lactose, and in this, it's a inducible operon. Why it is an inducible operon? Because this operon will work only in the presence of a inducer. So it's an inducible operon. So this is a I gene, which is a regulatory gene. It produces the repressor, right? So when there's absence of inducer, when the lactose is not present in the medium, in that case, this repressor will bind to the operator. Once this repressor will bind to the operator, it does not allow the RNA polymerase to bind to the promoter. If the RNA polymerase will not bind to the promoter, the transcription of the structure gene will not take place. So in the absence of inducer, no transcription is observed. But when there's a presence of inducer, when lactose is present, in that case, the lactose or the inducer will bind to the repressor. And it will lead to the inactivation of the repressor, because when the inducer bind to the repressor, it lead to some conformation changes in the repressor. Because of those conformation changes, the repressor cannot bind to the operator. Okay, if the repressor will not bind to the operator, now that means the RNA polymerase can easily bind to the promoter. When the RNA polymerase will bind to the promoter, it will lead to the transcription of the structural gene, which are Z, Y, and A. So in this case, we will observe the transcription of the structural gene. Z codes for beta-galactosidase. Y codes for permease enzyme. A codes for trans-acetylase. Remember this, that which structural gene is coding for the which enzyme. It's very important. Many times this question came in NEET. Okay, remember this. Z code for beta-galactosidase. This enzyme will act on lactose and it will break the lactose into galactose and glucose. Permease enzyme, it will make the cell permeable for the lactose. Third one is trans-acetylase. The function of trans-acetylase is to transfer the acetyl group from the acetyl-CoA to the beta-galactosides. Moving next is the Human Genome Project. The main objective of the Human Genome Project is to sequence or map the entire genome of the human. There are some silent features regarding the Human Genome Project, that a human genome contains 3164.7 million base pair. Every gene sequence consists of 3000 base pair, and the dystrophin, which is a human gene, consists of 2.4 million base pair. Total gene is estimated as about 30,000. Less than 2% of the genome codes for protein. That means less than 2% is the coding sequence, which are coding for the proteins. Repeated sequence, they make up the large portion of the human genome. Chromosome 1 has most gene, which is 2968, and Y has fewest gene, which is 231. Please remember this. These are very important. What are the salient features of Human Genome Project? Next one is the DNA fingerprinting. We know that the human genome, it consists of the large portion of the repetitive DNA, right? And we also know that these repetitive DNA, they are non-coding in nature. That means they does not code for any functional protein. These repetitive DNA, they are also known as satellites. And on the basis of their length, their segment, and the composition, and their number of repetitive units, there are two types of satellite. There are mini satellite and micro satellite. You can see in the figure, there's a mini satellite and this is a micro satellite. Mini satellite is also known as a variable number of tandem repeats, which is VNTR. Micro satellite is known as a short tandem repeats. In mini satellite, the repeat units, they are 100 base pairs. Okay, you can see that the length of the repeat units, they are larger in case of mini satellites. But in case of microsatellite, they are small. And here the repeated units are 4. 1, 2, 3, 4. But here the repeated units are 8. Here it is 20. So on the basis of the length of the segment, on the basis of the number of repeats, and on the basis of their composition, the satellite, they are of two types, the mini satellite and microsatellite. And we know that these non-coding sequence or this repeated DNA, they show a high frequency of polymorphism or a high degree of polymorphism. Polymorphism is the change in the sequence of the genome. When there's a, for example, if this is a sequence, this is a repeated unit. This sequence is repeating again and again. AG, 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 AG. This sequence is repeating. If this A get replaced with C, for example, if the change has occurred in this at A base pair and it is replaced by C, now the sequence has become AG, GC, AG, AG, right? So the change has occurred at this position, at the third position. If this change sequence spread in the population, if this change sequence is spread in 1% of the human population, which we call it as polymorphism. And that this change could be occurred due to mutation. So this is the basis, this degree of high degree of polymorphism is the basis for DNA fingerprinting. And we know that these polymorphism, they are inheritable. They can pass from the parents to their offspring. So we can also use DNA fingerprinting as to resolve the paternity disputes or for paternity testing. DNA fingerprinting was first or initially developed by Alec Jeffrey. He uses a VNTR, which is a variable number tendon repeats. And we know as the VNTR, they are mini satellites. We know that the 99.9% of the genes are same in all humans, right? But the variation lies in 0.01%. It's because of these repeated sequence, because they show a high degree of polymorphism. And these VNTR, for example, this is a chromosome 1, this is a paternal, and this is a maternal chromosome. So paternal chromosome is showing the 2 repeats of the VNTR. And the maternal chromosome is showing the 3 repeats of the VNTR. And this is same. This is a chromosome number, let's say 7. So you can see that there's a difference in the repeat units. And the paternal chromosome has 2 repeat unit and the maternal chromosome has 3 repeat unit. So the VNTR, their repeats, they can vary from chromosome to chromosome in an individual. Okay. Like you people also have repeated DNA. I'm also having a repeated DNA. But what the difference? The difference lies in the repeat units. For example, in your case, it would be a 4 repeat units. In my case, it would be a 6 repeat units. So difference lies in the repeat units. This figure is showing you a southern blotting. First, we extracted or isolated the genomic DNA. Now this genomic DNA will be subjected to the restriction endonuclease. In restriction endonuclease, they will work as a molecular scissor. They will cut the genomic DNA into the fragment. Now these fragments, they are placed on the agarose gel And the electrophoresis will be carried out where the separation of the DNA fragment on the basis of the size. Once the DNA fragments they are separated, they will transfer to a nitrocellulose or the nylon membrane. When they are transferred to the nylon membrane, now this VNTR probe are used. When they subject this membrane with the VNTR probe, we know that it is complementary. Okay, so once it get the complementary base pair, it will bind to the complementary DNA and it form a hybridized DNA segment. Now we can, while carrying out the autoradiography, we can detect this hybridized DNA fragments. You can see in this image, there's a chromosome number 7, and this is a paternal chromosome, and this is a maternal chromosome. This is also paternal, this is maternal. You can see the variation lies, or the difference lies in the repeat units. In the repeat, here it is 3, here it is 1, 2, 3, 4, here it is 1, 2, 3, 4, 5, 6, 7, 8, 8. It is 1, 2, 3, 4, 5, 6, 7. You can see this. There's a variation or the difference in the repeat unit lies in it varies from chromosome to chromosome in an individual. So this is a DNA from the chromosome from the individual A. This is a DNA from individual B. And this DNA is obtained from the crime scene. This is the DNA of the suspect, which is obtained from the crime scene. When they carried out the jelly electrophoresis, they see that this DNA, which is obtained from the crime scene, which is the DNA of the suspect, it is similar to the DNA of the individual B. Because every individual has a different DNA. Okay. So we can easily, from this figure, we can also easily make out that this DNA, which is obtained from the crime scene, is matching with the DNA of the individual B. So that means the individual B is the suspect. Now let's discuss the question. Question number 1 says, complete the flow chart on the central dogma. Options are given. Quickly answer what you think, what will be the correct option. The A represents replication. Now the information from the DNA will be transferred to mRNA. This process is known as transcription. Now this mRNA will be converted to protein. This process is known as translation. Very good. Yes, the third is A replication, B transcription, C translation, and D protein. So the correct option is 3. Question number 2 says, in lac operon, Z gene codes for. I told you, please remember that ZYA, they codes for different amino acid. Please they codes for different enzyme. Please remember their name. It's very important. Many times this question came in NEET. Yes, very good. They code for beta galactosidase enzyme, which will act on lactose and will break the lactose into galactose and glucose. Second one, the Y, we code for permease, and A code for transacetylase. Moving next is which of the following is required as inducer for the expression of lac operon? Which of the following is required as inducer for the expression of lac operon? Option 1 is glucose, option 2 is galactose, option 3 is lactose, option 4 is lactose and galactose. Yes, very good. It is lactose. Lactose will act as an inducer. There's one more important point which I have to tell you. We know that this lac operon, it is present in prokaryotes, right? So if the medium is having glucose, in that case, this lac operon will not be active because now the cell which are present in the medium, they can utilize the glucose. But when the level of glucose depleted, in that case, the cell will start using lactose. Let me explain it again. That lac operon, it present in prokaryotes, right? If the medium is containing, let's say it containing both glucose and lactose are present in the medium, the cell E. coli, for example, the cell here are E. coli. These cell will first consume the glucose. Their first preference would be glucose. When the level of glucose depleted, now this cell will use lactose as their energy source. Okay, when the glucose level depleted, now these cells will use lactose as their energy source. And in that case, the lac operon, now the lac operon will get activated because this lactose will act as an inducer. Okay, I hope it is clear. Thank you all for your participation. We are winding up the class. Thank you all. See you in the next class and have a nice day.